Hello and welcome to my channel. In this video we will discuss about reverse transcriptase polymerase chain reaction, or simply Rt-PCR. Our world is struggling today. Thousands are died, millions infected, billions in fear. COVID-19 has changed the world. All humans collectively work to fight the pandemic. All hope is not lost. Years of research and hard work by brilliant minds worldwide have provided humanity with the hope of survival. Drugs are being researched, vaccines being designed, centers are elaborated, and all are dedicated to minimize casualties. You might have heard the diagnosis of COVID-19 infection is being made within 8 to 10 hours with high accuracy. All this is possible because of a technique, although not new, but the whole world has understood its importance. This technique is called Rt-PCR. All living cells have genetic materials, which is a set of instructions of how the cell needs to perform its function, how it will grow, and how eventually it will die. Genetic materials can be found in two forms. The first form is RNA, which is ribonucleic acids, and the second form is DNA, which is deoxyribonucleic acids. Now, DNA is the genetic materials which is made of two strands found in all organisms, such as the animals, the plants, and some viruses. Whereas RNA is a single-strand molecule, it does some functions as compared to DNA. Polymerase chain reaction, or PCR as we know it, is a method which is widely used in molecular biology to rapidly make millions to billions copies of DNA. Scientists take a very small sample of this DNA to amplify it into large amount in order to study it in details. An American biochemist, Carey Mullis, invented the PCR. Now, some viruses, such as coronavirus, can only contain RNA in its genetic material. Viruses rely or depend on human healthy cells in order to survive, reproduce, and make several copies of themselves. Once in the cell, the healthy cells now get controlled by the viral cells and produce a lot of cells that are infected. This will produce problems with the traditional PCR because, as we know, in PCR only DNA can be detected, and coronavirus contains the genetic material RNA. How this problem is solved? In order for the virus, such as coronavirus, to be detected by the PCR machine, scientists need to convert the RNA into DNA by a process known as reverse transcription. So, as we know that copying DNA into RNA is called transcription, now the reversed method or technique will be called reverse transcription. So, using the technique of reverse transcription with the technique PCR is known as reverse transcriptase PCR, Rt-PCR. We use an enzyme called reverse transcriptase in order to convert the RNA into DNA. And PCR by now can make millions of copies of this viral DNA in no time. Scientists then will confirm if the virus is present in the sample or not. So now, how does real-time PCR work with the coronavirus? A sample is collected from the parts of the body where the virus is gathered, such as the nose and the throat. This sample is then added to chemical solution to remove fats and proteins and extracts only the RNA present in the sample. Now in the sample, this RNA extracts is a mixture of the person's genetic material and, if present, the coronavirus RNA. So now we have the viral RNA. This viral RNA is reverse transcribed into DNA using a special enzyme known as reverse transcriptase. From here onwards, the process is similar to PCR. So the aim will be a minute of genetic material DNA will yield several copies of that DNA. So how can we amplify this DNA into several copies? We can do so by PCR technique. So here is the DNA. The first step is to add specific short fragments of DNA to the sample. These fragments attach themselves to a unique targeted section on the viral DNA. They are just going to bind to a targeted section on the viral DNA if the virus is present in the sample. Additionally, the fragments also contain marker labels which are used then to detect the virus. The marker labels are going to give a fluorescent color that indicates that the sample is viral. Then the mixture is placed in a real-time PCR machine. Machine cycles through temperatures that heat and cool the mixture to trigger specific chemical reactions that create new identical copies of the targeted sections of viral DNA. Cycle repeats over and over again to continue copying the targeted section of the viral DNA. Now, how this happens in PCR machine, I'm going to explain in details in another video. Each cycle doubles the previous amount. So 2 copies become 4, 4 copies become 8, and so all the growth becomes exponential. Standard Rt-PCR usually goes through 35 cycles. So by the end of the process, 35 million copies of DNA sections are generated. As new copies of viral DNA sections are made, the marker labels release a fluorescent dye, which is a signal measured by the machine computer and presented in the real time on the screen. The computer tracks the intensity of the fluorescence in the sample after each cycle. When the amount goes over a certain level of fluorescence, the presence of the virus is confirmed. Scientists also monitor how many cycles it takes to reach this level in order to estimate the severity of the infection. The fewer the cycles, the more severe the viral infection is. And at the end, it's very important to list the benefits of RT-PCR. RT-PCR is highly sensitive, it's highly specific. It takes 3 to 4 hours to diagnose the virus, and some machines take an average of 6 to 8 hours. It means it's the faster method used, and it has a lower potential for contamination since it is used in closed tubes. PCR continues to be the most accurate method for detection of coronavirus yet. So this was all about RT-PCR. I hope you liked the video. Wait me for more videos. Thank you.